Okay. All right, our next one here is we're going to look at viral vectors. I'll type in viral vectors and gene therapy. And we get a whole bunch of them. I just typed this in to see what you get. You'll notice that we get a lot of adenovirus vectors. We also get adenoviruses. We get adeno-associated viruses, which are viruses which don't replicate on their own. They have to have another virus, such as an adenovirus, to help them. And we're also going to, well, and here we're just getting this, but we're going to look specifically now on gene therapy and adenoviruses. So I'm going to pop this in. Adenoviruses, and then we're going to look at lentiviruses in just a moment. So the idea with an adenovirus is that you are able to you're going to have inside of this viral DNA, but also a gene which is carrying whatever you want it to carry to go into the host. And the adenovirus has all the receptors necessary to be carried all the way into the host, and then also all the way into the nucleus. And so that's the attractiveness for that. And so it is going to bind. Here we have this CAR receptor right here, an integrin, and these knobs on these fibers for the adenovirus will bind to those. And also for AAV, we have it's a naked virion which has no fiber tails on it. It's an icosahedron, but there are specific proteins at the vertex of this icosahedron, of each of the pentamers here, and they will bind to a specific receptor as well. And then, of course, for the retroviruses, we have glycoproteins in the envelope all the way around here. It's just showing 2 for right now, but the SU and the TM, the transmembrane and the surface proteins, and they will bind to specific receptors as well. So if you get these receptors, these things will take you to specific cells. Okay. Now let's look at what happens when we get there. I had this last night. Oh, these are lentivirus vectors. Not yet. Cover that in a moment. Okay. This should help. So what we have then is we have some DNA which is placed in the viral gene, and this goes through by endocytosis, and it will disassemble and deliver the DNA right to the nucleus. And at this point, this can be incorporated into the nucleus, and because that's what adenoviruses do. And once it gets incorporated into the nucleus, then you have changed your cell, you have transformed your cell, and you can use that for gene therapy. So in other words, you can treat via gene therapy and actually change the cells this way. We had a wonderful graphic up here. Let me see if I can get it. I think it was under lentiviruses. So let me see if I can pull that one down. I can go to lentivirus vectors then. Yeah, here we are. So in this case, we have this person suffering from, this child suffering from a gene deficiency, a genetic deficiency. So what you do is you collect the cells from the, and these are the hematopoietic, in other words, stem cells. So these are the bone marrow cells, hematopoietic stem cells, HSCs. And they are the ones that will create the blood cells, so the bone marrow cells. You isolate those, and you go ahead and take this HIV-based vector, and that will add DNA right to the into the genome of the cells you've collected, and then you put those cells back into the patient. So that's the idea of doing this ex vivo, taking the cells out and putting the cells back in, gene therapy. Okay? So lentivirus vectors or adenovirus vectors. And if we look at a lentivirus vector, so we have, and you're familiar with this, 2 RNAs in here, and then we're surrounded by a nucleocapsid and then a matrix, and nucleocapsid, then a matrix, and then the membrane, and then these envelope proteins. A typical HIV, or a typical retrovirus. And so from here we get infection, and we have reverse transcription of the RNAs, and then these become double-stranded DNA, and then we have integration into the host. Okay? So that's another, just very quickly, we haven't gone in much, the idea of a packaging cell line. I guess we have to look at that also. Let me put that in. Lentivirus and packaging cell line. Let's see how that works. Okay. Oh, it's very small. Okay. That's bigger. Okay. So here, what we have is we have the actual construct, and for this one we have. I don't want that one. Sorry, I wanted RRE on that. Here we go. Okay. This gives us part of it here. So here's the lentivirus genome, and we have long terminal repeats on both ends. The long terminal repeats are what is grabbed by the integrase, and that's what is inserted into the recipient genome. GAG and POL and N are the genetic units, the main genetic units. There are all these other ones as well. And they are the ones that code for example, this one codes for nucleocapsid and capsid and matrix. This one codes for most of the enzymes. This codes for the envelope. And then we have other ones as well. Rev is important with HIV because what Rev does is it enables the longer longer transcripts to leave the nucleus and go into the cytoplasm. Because normally there's a very strict rule when you export RNA. You have to be fully spliced in order to leave the nucleus. So what Rev does is it enables you, it was a protein which will bind to sequences which are not fully spliced, which are actually fairly long, and it will pull those and allow them to be exported into the cytoplasm. If you don't have export into the cytoplasm, then that means you have no translation. So that's what Rev does. And Rev binds to an entity on the RNA called RRE, or the Rev response element. And so you have to, when you have your transfer plasmid right here, we have 2 LTRs, we have a cDNA, and then we have a promoter which can help that cDNA. And we want to transfer this. This is the part that we want to transfer into the patient, into the cells. We also have an RRE here usually, so that we can have efficient transfer of the entire mRNA out into the cytoplasm. But what we do is we mix this transfer plasmid, we put it into one cell, and we have the transfer plasmid and also a packaging plasmid, which contains the GAG, POL, TAP, and REV, and the others, and also another plasmid which carries N. We split this up, but we put a packaging signal only on this transfer plasmid. Only this transfer plasmid gets a packaging signal. So if we grow this in a cell, in your packaging cell line, which contains all these other elements, then we get 3 mRNAs being produced. This one, and this one. However, the only one with a packaging signal is this one. So all of these proteins are made, which will create the virion, which will create the virus particle, but only the transfer plasmid gets encapsulated. The other ones lack a packaging signal, so they stay behind. So your producing cell line will produce only virions which contain this, and that's what goes into the patient. So now, since this is all absent, this cannot replicate in the patient as a retrovirus, because it's missing all of these genes. Does that make sense to you? So if we separate the live virus into 3 different pieces, every piece is it cannot enter a cell, replicate, and form infectious virions. These stay behind because they don't have a packaging signal on them that the proteins can recognize for packaging. This is the only one with a packaging signal. That's the only one that gets made into a virus particle. That's the one that can go into the patient's cell. But it is lacking GAG, POL, N, and all these other ones. So it is unable to infect the patient anymore. What's the one drawback, you'd think, for lentivirus therapy? What can you think about? A drawback. You're going to take viruses and you're going to randomly insert them into your patient's DNA. What could be the problem? Have we already talked about this? We did, didn't we? So what's the problem? Claire, you got it last time. Why don't you get it this time too?
If it gets put next to a promoter, or a promoter turns on a cancer gene.
Cancer gene, right. And so that's the big problem with lentivirus vectors. Yeah. Okay, I forgot. I'm teaching the Molgen class. I forget what we covered. When I teach 2 classes at once. Okay. Let's move on then, and we're going to talk about vaccines. And I'm going to stop this one.